The vibrant coloration of the coral animal is actually the pigmentation of algae called zooxanthellae that reside within the coral's tissue. The maintenance of the coral-algal relationship is essential for the well-being of both organisms. Zooxanthellae residing within the coral tissue use nutrients made available by the coral. In return, the coral gains energy-rich organic carbon created by zooxanthellae through photosynthesis. This energy from the organic carbon is important for the production of the limestone coral skeleton. The loss of this algal inhabitant can be lethal to the coral animal. The reduction of zooxanthellae, or pigment concentration, is called coral bleaching as it exposes the white skeleton of the coral animal. A number of factors cause coral bleaching. One such factor is increased solar irradiance. The anthropogenic input of chlorinated fluorocarbons has significantly decreased the ozone layer during the second half of the 20th century. The result has been an increase in the flux of harmful ultraviolet UV radiation. UV light has a short wavelength and high energy that can be damaging to cells. Solar irradiance is known to affect zooxanthellae growth and photosynthesis. The coral animal has several adaptive strategies to help protect itself and its algal inhabitant from this deleterious wavelength. One proposed strategy is the possession of compounds homologous to green fluorescent proteins, GFPs, that are able to capture and dissipate the high energy from the UV light and re-emit it as non-damaging light at a greater wavelength. This effect can be seen by the fluorescent coloration emitted by the coral animal, captured with a camera equipped with a yellow filter when shined with a UV light. Another defense is the placement of different types of zooxanthellae among and between coral colonies that correspond to irradiance exposure. Zooxanthellae are single-celled algae of the genus Symbiotium that are genetically divided into distinct groups called clades. It is well established that tolerance levels to solar intensity distinguish each clade. Certain clades are shade-adapted compared to tolerant clades of high irradiance. As a result, clades can be spatially separated within a single colony and between different colonies of the same species. In shallow depths, for instance, zooxanthellae belonging to the shade-adapted clade C are in the lower portions of the corals Monastrea favulata and Monastrea annularis. Clade A and B are found in the top portion of the coral colony where irradiance is higher. These localized gradients of zooxanthellae clades and irradiance were maintained at greater depths. A possible explanation for the conspicuous bleaching patterns in a coral colony is the susceptibility differences to solar intensity. The top part of the colony may be exposed to an acute episode of greater level of UV radiation than their symbiote can withstand, which results in bleaching. Of the numerous factors that cause coral bleaching, high sea surface temperatures is best documented. Thermal stress is known to damage the photosynthetic apparatus of the zooxanthellae. However, recent investigations have shown that the elevated water temperatures can make the coral animal susceptible to microbial infections. Viruses and bacteria are ubiquitous and abundant in the marine environment. Scientific investigators Dr. Eugene Rosenberg and colleagues were able to prevent the thermally stressed coral Oculina panagonica from bleaching by treating the corals with antibiotics. Subsequent studies showed that the opportunistic bacteria Vibrio choloi is a causative agent. This bacteria normally resides on the surface of fireworms that feed at the base of the coral colonies. Increased water temperatures create extra surface proteins in the coral mucus that allows this pathogen to switch hosts. Once the coral is infected, the Vibrio pathogen releases a chemical compound that blocks photosynthesis or kills the zooxanthellae. Other studies by Dr. Wilson at the Plymouth Marine Laboratory revealed the presence of virus-like particles in the thermally stressed zooxanthellae. It is hypothesized that the elevated water temperatures induce these viruses, which kills the coral's symbiotic algae. The expected rise in sea surface temperatures may serve as a catalyst for opportunistic infections on coral reefs. CO2 concentrations from the burning of fossil fuels have dramatically increased in the Earth's atmosphere in the last 40 years. At the present rate, CO2 concentrations will increase twofold by the year 2100. Carbon dioxide traps the heat transmitted to the Earth by the sun, which is called the greenhouse effect. Conservative estimates, based upon current trends and the mildest greenhouse scenarios, project a drastic increase to around 32 degrees Celsius in sea surface temperatures during the next 90 years. Empirical evidence shows that mass bleaching events can be triggered by an increase as little as 1 to 2 degrees Celsius. For example, Corals belonging to the genera Acropora and Parietes typically bleach around 29 to 30 degrees. Sea surface temperatures reaching in the low 30s is lethal enough for mass mortality of these corals. The frequency and intensity of coral bleaching events and mass coral mortality has dramatically increased in the last several decades, providing a strong correlation between increased CO2 and coral bleaching. The global coral bleaching event in 1998 is the most severe in recorded history. A prolonged El Niño southern oscillation caused global thermal anomalies to increase sea surface temperatures as much as 3 to 4 degrees Celsius in certain regions. For the first time, almost every region in the world was affected with coral bleaching. The magnitude and severity of bleaching resulted in significant coral mortality. For instance, extensive mortality of the dominant Acropora corals in the Great Barrier Reef caused a major change in community structure. Surviving corals varied. This difference in survival is most likely the function of genetic variation in the associated zooxanthellae. Analogous to solar irradiance resistance, certain zooxanthellae clades can withstand higher water temperatures. Consequently, corals potentially expel susceptible zooxanthellae to gain thermally tolerant genotypes. This form of adaptive bleaching is a proposed mechanism of survival to increase sea surface temperatures. Studies indicate that the thermally tolerant clade D is more abundant on reefs following severe bleaching. In Panama, the ecologically dominant coral Pocillopora was sampled in 1995 and found that 43% of the colonies contained clade D and were unaffected by bleaching in 1997, while colonies containing clade C bleached severely. Returning in 2001, the percentage of colonies containing clade D had become dominant with 63%. Coral colonies in Kenya that were severely bleached in 1998 contained a high percentage of clade D in 2001, contrasting with colonies in Mauritius that avoided bleaching. This comparison was repeated between the Persian Gulf and Red Sea with similar results. What is of immediate concern is the expected rise in global temperatures and how this will affect coral reefs. Will increased sea surface temperatures result in the mass mortality of corals, or will corals have the ability to withstand and acclimate? The answers still remain ambiguous.